Ensembl, and my name is Alina Mushtaq, and I will be delivering today's module on Ensembl regulation. So let me just start with pasting the training page on the chat. And this is the link to the training page for the Nextgenhelper course. And on this link, you will be able to find the course book and the PDF version to my presentations, which includes all of the presentations that I've done this week, including today. And it's also going to have the presentation for Monday, the 26th of July. We also have a link to the shared Google Doc. It's called the Living Doc. And over there, you have a specific space to ask questions. Or if you get stuck somewhere, if you can't hear me properly, or my screen is, you would like me to zoom in, or if you're facing any technical difficulties or you are just not following me, please do let me know on the chat and I'll try my best to accommodate that. So it's now time for us to start. So let's start by talking about regulation of gene expression first. So we'll start by giving you a little bit of background. So if you take a look at the human body or any complex organism, you will realize that we are composed of hundreds of different cell types, and essentially all those cells carry the same genome and the same genes. But how come they're all so different then? This is because of epigenetic changes. So these are the changes to the DNA modification and DNA methylation, which result in cell differentiation. It is often irreversible, and as a result, these cells have different gene expression and they have clearly distinct morphology and phenotypes. And they all serve different functions as well. So ensemble regulation is the annotation of the genome with features that may play a role in the transcriptional regulation of genes. So these are things like gene promoters, gene enhancers, transcription repressors, and etc. And these are predicted using few main data types. So we use predicted open-close chromatin, we use transcription factor binding sites, epigenetic marks, so these could be histone modifications or DNA methylations, and also RNA polymerase binding. So let's imagine a gene model with this promoter. And for this gene to be transcribed, it needs to bind to a polymerase. So this is over here, this pink shape. However, before the polymerase binds there, it needs some other protein that encourages it to be there. And these proteins are called transcription factors. And there might also be additional proteins like activators binding to the enhancers, which kind of promote this conformation change of DNA and cause DNA bending and cause them to bring them near to a gene promoter. And it all creates a protein complex. It encourages the polymerase to bind there. There might also be insulators, which are genetic regions. Oops, sorry. I clicked too fast. So there might also be insulators, and these are genetic regions which repress transcription. So they bind CTCF, and this is shown as this green shape on my slide. So CTCF is transcription repressor. And there's also some histone modifications and methylations, which also affect the probability of protein binding at all of these sites. The recruitment of the protein, so the transcription factor, to the regulatory features, such as the promoters and enhancers, can be determined by a number of different factors. So we've already briefly touched on DNA methylation, and this is also shown in the diagram on my slide. But just to summarize, on the right-hand side, there are a number of different factors that can influence the binding of transcription factor to the DNA. So we can firstly talk about the chromatin conformation. So in this diagram, we have a chromosome which is formed of this chromatin being wound around and condensed into the chromosome. And this diagram shows this occurring in a very uniform manner. But in fact, the condensation of the chromatin is actually a very non-uniform process. So usually what happens is that there are regions of the chromatin that are very densely packed, like we might see here. And this is associated with transcription inactivity. So where the chromatin is tightly wound and condensed, the transcription factors are less able to access the DNA motifs within the DNA sequence. But then there might also be regions that are less well packed into the chromatin. So in this case, they might be much more open and accessible for the transcription factors to bind to the respective motifs. Another important thing that affects the accessibility and the binding of the transcription factor proteins to the DNA motifs is the positioning and the modification of the histone proteins themselves. So DNA, as part of its packing and condensation, is wound around proteins called histones. And histone proteins themselves are quite globular, and they have an N-terminal tail, which extends away from the protein itself. And these N-terminal tails of the histone can be post-translationally modified. So you see here on my slide. So these could be post-translationally modified with a number of different post-translational modification groups, such as acetylation, methylation, ubiquitination, and so on. And these different modifications can then influence the recruitment of the transcription factor to the specific motifs and therefore affect the transcriptional activity. There are a wide array of experimental techniques that scientists can use to explore the different levels of transcriptional regulation that is present across the different regions of the genome. This image is taken from the ENCODE project, and it's describing the different ways that we can assay the transcriptional regulation at these different levels. So firstly, we have the chromosome conformation over here through combination of 5C and high C experiments. Then we can have regions of open and closed chromatin, so whether they're open or closed, like it's shown over here. And we can use techniques like DNA-seq or FAIR-seq and ATAC sequencing. We can also look at a number of different things with an experiment called ChIP-seq. So this is a chromatin immunoprecipitation followed by sequencing. And what this does is that it allows for us to look at the proteins that are interacting with the DNA. So these proteins might be transcriptional factors themselves. By using ChIP-seq, we can experimentally assay to see where along the genome the different transcriptional factors are binding, but we can also use ChIP-seq to interrogate the different histone modifications that we can observe across different regions of the genome as well. We can use antibodies in our ChIP-seq experiment that bind specifically to the histones that are modified in a particular way, and use that followed by sequencing to find the type of histone modification that we observe in a given part of the genome. Then we can also look at things like DNA methylation by using whole genome bisulfite sequencing, or reduced representation bisulfite sequencing, or methyl arrays. Then we can look at the gene expression, so we can look at computational predictions, RNA sequencing, and we can actually look at the transcriptional efficiency by using ClipSeq and RipSeq techniques as well. So this is just a summary to show you that there is a wide array of experimental techniques that are used to interrogate the transcriptional regulation that's occurring across the genome. And now we're going to talk about how Ensembl can make use of all of this data from all these different types of experiments that are being generated by different consortiums and groups all around the world, and how we use that to annotate regulatory features across the genome. So we'll go through the different steps involved in Ensembl regulatory build. In step 1, we take raw data from a number of different sources. So there are different collaborative projects that have looked at a number of different cell types for human and mouse, and we take that raw data from these different experiments for the different cell types. Then for step 2, we process this raw data. We align it to the genome to predict the position of the regulatory features, and we assign the regulatory feature types. So these are feature types such as promoters and promoter flanking regions, enhancers, CTCF binding sites, open chromatin regions, and transcription factor binding sites as well. The third step is to then predict the activity of these features in the different cell types that we have experimental data available for. So the activity that we assign might be active, poised, repressed, inactive, or unknown. And then in step 4, finally, we display all of this information through the Ensembl genome browser. We input raw data from various sources. So we have ChIP-seq experiments and histone modifications, and DNA's hypersensitivity for human from Blueprint epigenomics, ENCODE, and Roadmap epigenomics. And for mouse, we input data from ENCODE. We then align the raw data to the reference. So this is shown over here as this blue line. And we call peaks from the reads coverage after specifying a cutoff. So we have these signals that represent the reads coverage, and wherever you can see the coverage is high, we would call a peak. We then have feature predictions. So we predict the position of regulatory features like for promoters, enhancers, and insulators. Once we have called our peaks from our raw read, you can see that we can find these different clusters of peaks scattered across the genome. And then we ask the question that, do we know what any of those peaks do? Because the chance is that if there are some known promoters which map to some of these clusters, the other clusters are also promoters. And we would do the same for other clusters as well. The next step is segmentation. So we would move along the genome to segment the genome into regulatory regions or not. We would look at these clusters, which are called from the raw data across the different experiments, and then ask whether there are known clusters, so similar clusters of known function. We would then segment the genome, and we would assign labels based on cluster similarity. So our segmentation algorithm is blind. It has no idea what the patterns are that they're looking at. It's all about searching for the patterns. So we're looking for similar patterns of known function, and based on that, we assign their regulatory predictions to other clusters of peaks. We then predict the activity of these features in the different cell types that we have available in Ensembl. So what we do is that we take the regulatory feature annotation across the genome for all cell types. So we're using 4 here as an example. And then we move along the genome in 200 base pairs, 200 base pair blocks, asking at that position, are there any regulatory features annotated? So in this case, we would say yes. So we're here. So in cell type 4, part of this 200 base pair block has a regulatory feature annotated. So we annotate a regulatory feature over here. And we move along the genome in this 200 base pair blocks, asking the same question again. So here we can see another feature in cell type 1 and 3 and 4. And finally, we have our final feature in blue. So what we're doing at the top here is constructing our regulatory build. So this is our multi-cell regulatory features. And then the final step is to look at each of these cell types and ask, is there any evidence to suggest that these regulatory features are active, poised, repressed, or inactive? In this case, cell type 1 and cell type 4. There's evidence to suggest that these regulatory features are active, so they will be colored in red. Whereas in cell type 2 and 3, there's no evidence to suggest that they are active, poised, or actively repressed, so they are inactive, which is what we see depicted here. We will also see this in the demo further, and we'll look at the regulatory feature activity in different cell lines. So once we have segmented the genome into regulatory elements, we then can access the activity of these elements across the different cell types. So there are 5 main activity types across Ensembl. These certain elements might be active, which means that it has all the proteins consistent when active. So for example, like a promoter promoting. It might be poised, so it's sort of ready to be activated, but it needs one element which is missing, so it needs a trigger. Then there is inactive. This means that it lacks the protein expected for the activity, so nothing is going on. Or it could be repressed, so this means that it has a protein attached to it which represses its activity, so things which block transcription. And then finally, we might not have data for the feature at all. And then step 4, which is the last step, and this is where you can view the data in the genome browser. And this is what we're going to do in a minute. We will look at the different cell types. We will look for regulatory features and their activity within those different cell types on the Ensembl genome browser. But before I move on to the demo, I wanted to mention a few limitations. So we don't link regulatory features to genes. We tell you where those regulatory regions are across the genome, but you have to make inferences yourself. So the chance is that if there is a promoter next to the gene's 5' end, it is likely that the promoter is driving this gene's expression, but you have to make these inferences yourself, which you would then need to experimentally validate and verify. So there are some limitations to the data that you're looking at in Ensembl, and this is because this data is really, really complex. But we do hope to eventually link regulatory features to the genes that they control the expression of. So these are some of the other regulatory data resources that we have in Ensembl. So we have for human and mouse, we have Vista elements, the enhancers that come from the Vista project. We have annotated in Ensembl, so you can also view those in the browser. We also have some predictions of mRNA target from Tarbase, and then for humans only, we also have the phantom transcription start site and enhancer annotations, and also GTEx EQ12 data. Finally, we also have methylation data from the ENCODE project that you can add as tracks on the location view. And within the transcription tab, you can also view the microarray probes mapping of the different transcripts of your interest. Some words on future directions. So in the future, we hope to extend this technology to a number of different species. Right now, we have it for human and mouse. So there are a number of projects that are aiming to generate a similar type of data for different cells of different species. We have the FANT project, which is looking at agricultural species. And then there's also other projects like Aquafang, looking at fish species. And then there's also the Zebrafish Genome Biology Consortium that is looking at getting similar data for zebrafish. And one of the major goals that we have is to link the regulatory features to genes that they control the expression of. So we're looking at using EQTL data and also Hi-C data that is currently being produced, published, and made publicly available, which we will then be able to use in our annotation process. Okay, let's move on to the demonstration part now. So I will be looking at a region of the KPNA2 gene and find regulatory features in it, and also explore what cell types that they are active in and what evidence there is to show this. So I'm going to jump out of my presentation, and you can find the demonstration on your course books by following the link that I've pasted in the chat box, and you'll find the demonstration on page number 67. So let's go to Ensembl homepage, and we're looking for the KPNA2 gene in human. So I'm going to first select my species of interest, and now I'm going to type my gene of interest. Once I'm happy with my specifications, I'm going to click Go. I'm going to choose the first result. And I'm going to go to the location page, the location tab for this gene. So I want to scroll down and bring your focus to region in detail view. And this is the most detailed image. And I'm going to zoom out a bit just so that I can see the gene's flanking region as well. So I'm going to click here to zoom out. So over here, now we have the gene's 5' end. And if I scroll down, you'll see that we have the regulatory build track shown by default. So in this regulatory track, we have these regulatory features, and then we also have the legend for the regulation over here on the bottom of the image. So we have promoter color-coded as red, then we have the promoter flank region shown in the salmon color. We also have the CTCF shown in cyan. So I'm going to click on this promoter that is shown in red. And you'll see that once I've clicked on it, there is a pop-up box that appears that gives me information on this regulatory feature. It gives me the regulatory feature stable ID, the type of this feature, so it's a promoter, its genomic location, and some more information as well. I'm going to click on the stable ID, and this is going to take me to the regulation tab, and this is a tab that we haven't explored before. So we are now on the regulation tab, and we have some regulation-based displays shown here as icons, and you'll see the same displays shown here on the left-hand side menu as well. And then similar to other pages, we have the options to configure this page, custom tracks, share this page, or bookmark this page as well. And if I scroll down, now we have the graphical representation of this promoter. So we have our regulatory build, so we have our promoter here, and we also have, so the promoter here is shown as this red box, and then we've also got the promoter flanking region in pink, and then we also have this other track that shows us the transcription factor binding motifs, and it's showing you all of the transcription factor binding motifs that fall within this promoter, and these are shown as clusters, and you can see that we've got these gray boxes, these gray motifs, and also black motifs. So the black ones are the ones that are experimentally validated in at least one cell type. So I'm going to click on one of these black motifs. And this opens a pop-up menu as well to show me their stable ID, their genomic location. We also have the transcription factors, the names of the transcription factors that bind to this motif. And then you can also find the cell lines that this motif has been functionally validated in. And then we also have the binding matrix. So I'm going to click on binding matrix. And this shows us a nice pop-up window with a really nice colorful image of the binding matrix. And we also have its stable ID over here. So let's go back to our page. Close this. Then if I scroll further down, we also have a summary of the regulatory activity. So we have a summary table that lists all of the cells, of all of the cell lines that this feature is active in or inactive in, poised in, or repressed. So we can see that our promoter is, you can find the demo on page number 67 of your course book. We can also find the cell line that our promoter is active in. So it's active in astrocytes, and it shows you that it's active in 1 out of 118 cell lines. So let's explore this data in a bit more detail. So I'm going to scroll back up. And I'm going to click on details by cell type. So I'm going to go here by clicking on this icon. So after I've clicked on it, we don't see any data here. So the page isn't showing any data at the moment because we haven't chosen any cell types yet. So let's click on Configure Cell Tissue button. This is this blue button over here, and we'll add more data to this view, including different cells and experiments. So let's say that I want to explore this promoter in, let's just reset everything. Okay, so let's say that I want to explore this promoter in different cell types. I can search for it by either using this alphabet ribbon over here, or alternatively, I can find them using this search option here. So I'm going to search for this promoter in, let's say, astrocytes. And I'm going to turn this cell or tissue line on by clicking on it here. And then I also want to search it in aorta. So I'm going to search for aorta and select this as well. As soon as you select your cells, they're added to this list over here on the right. So it shows you that we've selected 2 out of 148 available, and you can expand on this list by clicking on this plus icon that shows you exactly what you've chosen. So now let's choose the experiments. So I'm going to click on experiments. And basically you can navigate and you can select your experiments in a very similar way to the cells and tissues. However, the experiments are divided into histones, open chromatin, and transcription factor. So for histones, I'm going to select all of the experiments. So let's just select all. And then for open chromatin, there's only one available. So let's select that. And then for transcription factor, let's select CTCF. So as you can see now that our experiments have been added to our summary on the right over here. So So we have histones and we've selected 9 out of 9 available. For open chromatin, there was just 1 available and we've selected that. And for transcription factors, we've selected 1 out of 2 available. Once we're happy with our selections, we can click on Configure Track Display, which is this green button here. So here we have a summary matrix of the cell types that we've chosen and all the experiments that are available for them at the top. And we can customize the view, so we can choose by clicking on this whether we want the data to be displayed as peaks or signals or both. So we're going to go for both. So I'm going to leave it as is. And once we're happy with our selection, we can simply click on View Tracks over here. This is going to take a bit of time to load. Okay, so now, apart from a regulatory build, you can also see activity of a promoter in the 2 different cell types that we've chosen. So we have iota over here, and then we have astrocyte over here. And you can see that our promoter is inactive in iota. So if we click on it, it gives us the stable ID, it shows us the type of the feature, and then it shows us the status. So this is inactive. And then we also have some motive information over here as well. So let's close this. And then for astrocytes, our promoter is active. So if I click on it to find more information, it shows us the type of our feature, the stable ID, and then we have the status that shows active. So as you can see on this page, that we have all of the experiments for both lines, for both cell lines, and we can see the peaks shown here as blocks, which were called from the raw data. So from the reads coverage, which you can see at the bottom as a signal. And you can see that for the experiments that we've chosen to display, the signatures of the histone modifications are different for the 2 cell lines. So we have different clusters of peaks, and this suggests that those cell lines have different epigenetic profiles. And this is all that I wanted to show you about the regulation data that we have in Ensembl and in my demonstration. You'll get to explore the regulation data a bit more in the exercises. So let's go back to the presentation. And you can find the exercises on page number 71. There are about 2 exercises, but they are long. So please do feel free to ask any questions if you get stuck anywhere, and I'll be happy to demonstrate them on the screen. And this module was a bit short, so we have a lot of time for the exercises. So let's say that we will stay here until the end of the session to give time for any of the questions or any questions related to the exercises. Okay, so there are a few questions about exercise R1. So let's just do that on screen. Let me just find the exercise quickly. So we have to find the location for the region between the genes INSEG1 and BLACE, and the exercise asks us if there are any predicted enhancers in this region. Okay, so let's go to the Ensembl homepage. And let me just, this is human genes. So we're going to select our species and we're going to search for INSEG1. So let's go back. I want to show you something quickly. So on our search results, we can either just click the INSEG1 human gene, and this will take us to the gene tab. Or if you want, you can also click on this location, so this genomic coordinates over here, and this will take you directly to the location tab. So now we're on the location tab, and we have to find the location between genes INSEG1 and BLACE. So BLACE is here and INSEG is here. So what we can do is that we can draw in this region over here. And now we can select jump to this region, which will zoom out this exact region for us. So let's just wait for this region to be zoomed in. So this is our region of interest now, and I'm going to scroll down to see our regulatory build. So this is our regulatory build, and the question asks us if there are any predicted enhancers in this region. So enhancers are shown, if you go to the regulation legend, enhancers are shown as these yellow boxes. And in our regulatory build, we do have this one yellow box. Part B asks us to go to the regulation tab for the enhancer. So let's just click on this enhancer. And then it gives us its stable ID, which is ANSR 00001133586. And we can go to the regulation tab by clicking on the stable ID here. So now we have this graphical representation of this enhancer. We also have the motif feature track, and if we scroll down, we can find a summary table of the regulatory activity. And I think the question has asked us, are there any cell types where its activity is repressed? No. So how many cell types is this enhancer active in? So we have this information here. It's active in 1 out of 118 cell types. And are there any cell types where its activity is repressed? So we can find repressed over here in this column, and it's repressed in 34 out of 118. And we have all the list of the cell lines mentioned below. So I hope this answers the question. Okay, so we also have a question on part C, which has asked us to take a look at the histone modifications across an enhancer in 2 different cell types. So let's just click on details by cell type. And we're going to go to configure cell and tissue. And let's just reset all, because this is, I remembered my configurations from before. And we're asked to look at the histone modifications in this enhancer in the nucleomyelocyte cells. So let's just, let's use the ribbon this time. So I'm going to click on N, and I can find the nucleomyelocyte cells here. And it has also asked us to turn on the neutrophil CB cells. So let's just turn on neutrophil CB cells as well. Now that we've selected our cells and tissue, this is also available on the right-hand side. We now have to select our experiments. So the exercise has asked us about histone modifications across this enhancer. So let's just select all of the histone modifications. So I'm going to click on select all. And now let's go to configure track display. And I want both peaks and signals over here. So I'm going to leave it as is. And I'm going to click on view tracks. So now we can see our experiments for the nucleomyelocytes and the neutrophil cell lines. And over here, we can see the histone modifications, histone modification experiments as well. And you'll see that the peaks are different for the 2 cell lines. So we have these, some of the experiments mentioned here that are different for the nucleomyelocytes. And I think this is all that the exercise is asked for. Let me just check. Okay, so the difference, and then we have to go back to the summary page. And are there any verified transcription factor binding motifs in this enhancers and in what cells? So let's go back. To summary, and we can go back to the summary by clicking on this summary hyperlinked text on the left-hand side. And over here we have these motif features. And as I mentioned, that these are clusters of gray and black means that these motif features have been experimentally validated. So we can click on these black motifs to find out more information about them. So if I click on it, it gives you the names of the transcription factors that the motifs bind to. And then we also have the name of the cell lines that these motifs have been experimentally validated in. I know that the answer for R1 part D says that there are 2 motifs that are verified in K562 cells and some others, but because, as I mentioned, that the Ensembl website gets updated with the new data, so the answers might sometimes be a little bit different, but the exercises are more there for you to explore the different data that we have in Ensembl, just so that you can realize where the different things are and how you can find the different data. So you can find the experiment's transcription factors by going to details by cell type. Then you need to go to configure cell tissue. And then you need to first select your cell lines by in the cells and tissue tab. And then within the experiment tab, you will find histone experiments, open chromatin, and transcription factor. But you can only select these experiments once you've selected your cell lines. So in exercise 2, let me just quickly check what this exercise is asking us. So I'm not really sure if you have to configure this page for the details of cell type for exercise 2. But let's just select all for this, and then for experiments, transcription factor, you can also select all of the transcription factors from here. And now I'm going to click on this green button that says configure track display. And I'm happy with this. And then I'm going to click on view tracks. So this is going to take a lot of time because I've selected all of the cell lines. So 148 out of 148 and all of the transcription factors available for it. But at least this shows you where you can find the different cell lines and the transcription factors in the experiments tab. So I've read the question again, and the question is asking us for CTCF binding sites. So let's go back to select tracks, and within this transcript, so on the experiments tab, and after choosing the transcription factors, let's start by deselecting all first. You can find the CTCF experiment over here. And then you can just click on view tracks. And now our page has loaded. So we have this CTCF binding track for all of the different cell lines that we have selected. So this image would be quite massive because we selected all of the cell lines. And then there is a question about how to go back to the original page. So do you mean the summary page where we have the graphical representation of our actual feature? So to do that, well, you can click on Summary on the left-hand side, or you can just click on the summary icon over here as well. And this takes us back to the original page. This is because the I'm on the Ensembl site, so for Sanger, but I can go to the Asia site. So let me just show you how. I'm going to click on Help and Docs on the blue navigation bar at the top. And from here, if I go to Mirror Sites, this is in the About the Ensembl project. So on the Mirror Sites, I can then choose to go to the US West, US East, or Asia. Sometimes Ensembl can be really slow depending on the traffic, and this is where you do have the choice to go to the different mirror sites, and depending on the time there, the traffic might be low and the website might be a little bit faster. I will navigate to the Asia site and try and do one of the exercises and get back to you if I can see similar features or not. And in the meantime,
Lena, I think your sound has actually temporarily cut out. I'm not sure if you can change your settings.
oh, okay. Now, I was just saying that in the meantime, I'm also going to try going on one of the regulatory features and try to configure these cell types. Because I've had this problem before, and sometimes you can get around it by using a different browser. So I'm on Google Chrome right now, but it might work for you if you go on Firefox, for example. Okay, so I'm able to select the cell types, and I'm on Ensembl Asia. So it might be worth just changing your browser that you're using. If you're on Chrome, switch to Firefox, and please do drop us a help desk so that we can look into it further.